Welcome to our deep dive into the most impactful longevity research from the past couple weeks, specifically for experts and researchers like you in longevity biotech.
Absolutely.
We've focused on studies from credible institutions that challenge the existing models and could reshape how we approach aging.
Looking forward to it.
Get ready for some aha moments.
Yeah.
So let's jump right in. First up, a paper published in BioRocks on January 27th by Martinez Garcia and others from the Universidad de Oviedo in Spain,
Okay.
titled Reduce Systemic Autophagy by Simultaneous Loss of ATG4B, ATG4C, and ATG4D Leads to Accelerated Aging in Mice.
Wow.
What makes this one so significant for our field?
This study provides some of the strongest evidence yet that autophagy is not just involved in aging, but absolutely critical for healthy aging.
Okay.
The researchers created a mouse model lacking 3 of the 4 ATG4 proteases, which are enzymes that are essential for autophagy.
Got it.
Think of it as the cell's waste disposal and recycling system.
Okay, so they basically hamstrung the cell's ability to clean up.
Exactly.
What happened to these mice?
Yeah.
The results were striking.
Yeah.
These mice experienced accelerated aging across the board.
Oh, wow.
They had genetic damage, systemic senescence, and significantly shorter lifespans compared to normal mice.
Wow.
It really challenges the existing model because it establishes a direct causal link between reduced autophagy and accelerated aging.
So does this mean that boosting autophagy could be the key to fighting aging?
Well,
What are your thoughts on the therapeutic implications here?
it's certainly a very promising avenue for intervention.
Yeah.
Imagine being able to enhance our cells' natural cleanup processes and delay the onset of age-related decline.
Right.
This study opens up a whole new world of possibilities for developing autophagy-enhancing therapies,
It's definitely a paper that will have people talking.
for sure.
Let's shift gears to another fascinating study published in MedRax4 on January 28th.
Okay.
This one's by Kuo and others from the University of Connecticut Health, and it introduces a brand new way to measure healthy aging, the Health Span Proteomics Score,
Okay.
or HPS.
This feels like a big step forward from simply measuring lifespan.
Absolutely. We've known for a while that lifespan doesn't always equal health span,
Right.
but quantifying healthy aging has been a challenge.
Yeah.
The HPS score could change that.
I'm curious, how did they develop this score? It sounds pretty complex.
They used data from the UK Biobank analyzing a whopping 2,920 proteins in over 53,000 people.
Wow.
These proteins, when taken together, paint a detailed picture of an individual's biological state.
So it's not just one protein, but a whole symphony of them.
Precisely.
This could be incredibly valuable for personalized medicine risk assessment and even for tracking the effectiveness of longevity interventions.
Absolutely.
Let's dive into another critical aspect of aging.
Yeah.
Immunity.
Okay.
We all know our immune systems weaken as we age, making us more susceptible to infections and diseases.
Right.
A paper in Nature Reviews Drug Discovery on January 29th by Bracken and others from University College London tackles this head-on.
Yes, this paper really pushes us to rethink our approach to age-related immune decline.
Okay.
Instead of tackling inflammation and cellular senescence separately, they argue for therapies that target the interaction between these two processes.
So instead of looking at them in isolation, we need to consider how they influence each other within our tissues,
Exactly.
a systems approach to immunity.
Exactly. It's a vicious cycle.
Yeah.
Inflammation drives senescence, and senescent cells, in turn, fuel more inflammation.
So what kind of therapies are they proposing that could break this cycle?
They discuss several promising options, including senolytics, which selectively eliminate senescent cells.
Almost like taking out the trash.
That makes sense.
What about xenomorphics?
Yeah?
I've heard that term, but don't fully understand how it differs from senolytics.
They're more like damage control.
Okay.
Xenomorphics modulate the harmful effects of senescent cells without actually killing them.
So they quiet down the troublemakers.
Exactly.
This targeted approach to boosting immunity in older adults seems very promising. Moving on. Let's turn our attention to NAD, plus another hot topic in longevity research.
Okay.
A paper published in NPJ Aging on January 28th by Shinichiro Imai from Washington University School of Medicine dives deep into the role of the NMN transporter in ENMPT in controlling NAD+ levels.
This is a must-read for anyone working on NAD plus boosting therapies.
Okay.
It emphasizes the crucial role of these two players in maintaining NAD plus levels as we age.
I'm thinking of it like a supply chain.
Okay.
We can pump in more NAD+, but if the transport system isn't working, it won't get where it needs to go right.
A perfect analogy. This paper highlights the importance of not just boosting NAD+ levels, but also ensuring that the necessary transport mechanisms are functioning optimally.
It's a good reminder that aging is complex with multiple interconnected factors at play.
For sure.
We can't just focus on one thing in isolation.
Right.
Now, for something truly captivating, a study published in Geroscience on January 27th by Chang-Hin Park's team from Iwo Women's University in Seoul.
Okay.
This one focuses on superagers,
Yeah.
those individuals who maintain exceptional cognitive abilities well into their later years.
Superagers are fascinating, aren't they?
Yeah.
They challenge our assumptions about what's possible with aging.
This study looks specifically at their brains using longitudinal data to compare them to the brains of typical older adults.
Interesting.
What did they discover?
What they found was remarkable. Superagers actually exhibit slower brain aging.
Oh, wow.
Their brains showed less shrinkage and maintained better structural integrity compared to their peers.
So it's not just that superagers are mentally sharper, their brains are literally aging more slowly.
That appears to be the case.
That's incredible. It seems like maintaining brain health is key not only for extending lifespan, but also for enhancing health span.
Absolutely.
Yeah.
This study further supports the idea that we have more control over our aging process than we might think.
Right.
Lifestyle factors like exercise, mental stimulation, and social engagement are likely playing a role here.
This is definitely an area that warrants further investigation.
Agreed.
What secrets do superagers hold for staying mentally sharp as they age? I'd love to know.
Me too. While we don't have all the answers yet, this research points us in the right direction.
Yeah.
It's exciting to think about the possibilities.
Now, before we move on to even more groundbreaking research, are there any reflections on these first few papers that you'd like to share?
What strikes me is that we're seeing a shift from simply describing the hallmarks of aging to really understanding the underlying mechanisms and identifying potential points of intervention.
Uh-huh.
It feels like we're entering a new era of longevity research.
I completely agree. Let's dive into some more of these exciting discoveries.
Sounds good. Speaking of exciting discoveries, let's delve deeper into the cellular machinery that drives aging.
Okay.
Remember those proteasomes we talked about earlier?
Yeah.
They're like the recycling centers of our cells, breaking down damaged or misfolded proteins.
Uh-huh.
A paper published in PubMed on January 27th by Hunt and others from St. Jude Children's Research Hospital really highlights their importance, especially as we age.
Right. They're essential for keeping things running smoothly.
Exactly.
What did this study reveal about proteasomes and their role in aging?
They focused on an enzyme called UBE2D, which is crucial for maintaining proteasome activity.
Okay.
The researchers discovered that as we age, UB2D levels decline.
So less UBD2D,
Right.
less efficient proteasome activity. What are the implications of that?
Exactly. It leads to a buildup of those damaged proteins we talked about interfering with normal cellular processes.
Right.
Think of it as the recycling center getting overloaded and unable to keep up.
Yeah, I gotcha.
This accumulation of damage contributes to a whole host of age-related issues.
Does this study suggest any ways to combat this age-related decline in proteasome activity?
What's really interesting is that they found that restoring UBE2D levels could partially reverse these age-related changes in flies.
Oh, wow.
It suggests that targeting UBE2D could be a promising avenue for developing therapies to improve proteasome function and combat aging.
It's amazing how these tiny cellular components can have such a significant impact on lifespan.
I know, it's fascinating.
It really highlights the interconnectedness of everything.
It really does.
Speaking of interconnectedness, a paper published in Biocoxturve on January 29th by Yukim and others from the Fred Hutchinson Cancer Center caught my eye.
Okay.
It delves into sequence editing and its role in regulating two key proteins involved in aging SKN1A and SKN1C.
This study provides some fascinating insights into how sequence editing, that process of fine-tuning proteins after they are made, impacts longevity.
Okay.
These SKN1 proteins are like master regulators of cellular stress responses, playing a critical role in protecting our cells from damage.
So how does sequence editing fit into all of this?
They found that sequence editing is essential for SKN1A to properly activate genes involved in producing proteasomes.
Okay.
So without the right editing, those crucial cellular cleanup crews can't function optimally.
It's like ensuring quality control for the recycling center.
Exactly. They also found that sequence editing influences the activity of SKN1C, which protects cells from oxidative stress.
Got it.
This suggests that sequence editing plays a multifaceted role in regulating cellular processes critical for longevity.
It's like a delicate dance happening at the molecular level to keep everything balanced.
It is.
Stepping back from the cellular level for a moment, Let's talk about how different organ systems communicate and interact with each other as we age.
That's a crucial aspect of aging that is often overlooked.
Yeah.
A paper published in Geroscience on January 28th by Orestes Stylianou and others from the Emmanuel Clinic Rudersdorf and University Clinic of Brandenburg Medical School tackles this very question using a fascinating approach.
They looked at whole body networks, or WBNs, right? It's like a systems biology perspective on aging.
Yes. They used functional magnetic resonance imaging respiratory rate and heart rate data to create these WBNs.
Wow.
It's a way to visualize the communication pathways between different organs.
And they found differences between the WBNs of young and elderly individuals.
Significant differences.
Oh, really?
Some connections were stronger in older adults while others were weaker.
Interesting.
It's like the communication pathways within the body are being rewired as we age.
That's a powerful image.
Yeah.
What are the implications of these rewiring patterns for our health and well-being?
They found fascinating correlations between these WBNs and cognitive function.
Okay.
Better interconnectivity and segregation within the WBNs were associated with better short-term memory and verbal learning in younger participants.
So maintaining healthy communication between different organ systems is crucial for preserving cognitive function as we age. It reinforces the idea that aging is a whole body phenomenon.
Precisely. This research opens up a whole new way of thinking about aging and could lead to the development of interventions that target these whole body networks. It's a truly holistic approach.
Now let's turn our attention to a topic close to everyone's hearts, literally.
Okay.
A paper published in bioRxiv on January 20th by Martinac and others from the Victor Chang Cardiac Research Institute explored the role of a protein called piezo-1 in cardiac aging.
Piezo-1 is a fascinating protein.
Okay.
It acts as a mechanosensitive ion channel in the heart, a sensor that responds to the mechanical forces of pumping blood.
So it's involved in how the heart adapts to the stress of its workload. What did this study reveal about its role in aging?
They deleted piezo-1 in adult mouse cardiomyocytes and observed accelerated cardiac aging and premature death.
That's significant. What specific signs of accelerated aging did they see?
The mice's hearts showed elevated markers of senescence, impaired cardiac relaxation, and developed sinus bradycardia, a slow heart rate.
So piezo-1 seems to play a crucial role in maintaining normal heart function as we age. Are there any clues as to how it does that?
They found a link between piezo-1 and the production of atrial natriuretic peptide, or ANP, a hormone that regulates blood pressure and has antifibrotic effects.
Okay.
Deleting piezo-1 reduced ANP levels, impairing the heart's ability to release ANP in response to stretching.
So piezo-1 helps protect the heart from fibrosis, the stiffening of heart tissue that often occurs with aging.
Exactly.
This could have important implications for developing therapies to prevent or treat age-related heart conditions.
Absolutely. And it underscores the importance of understanding these intricate molecular mechanisms in the context of aging.
Now let's shift gears to a topic that often sparks debate in longevity research: the relationship between reproduction and aging.
Okay.
Does having children speed up the aging process? A paper published in Metrics on September 23, 2024, by Yang and others from the Buck Institute for Research on Aging offers some intriguing insights.
This study used Mendelian randomization, a technique that leverages genetic data to assess causal relationships. They investigated the genetic associations between the age of menarche, the onset of menstruation, and the age of first childbirth with various aging-related outcomes.
So they were looking for genetic clues to understand how the timing of these reproductive events might influence aging. What did they find?
They discovered that later ages of menarche and first childbirth were associated with several positive aging-related outcomes.
Okay.
Longer parental lifespan, decreased frailty, and slower epigenetic aging.
It suggests that delaying motherhood could be beneficial for longevity.
That's what the data suggests.
Did they look at specific age-related diseases as well?
Yes, they found that later menarche and first childbirth were associated with a lower risk of developing several age-related diseases, including Alzheimer's disease, type 2 diabetes, heart disease, and COPD.
That's a pretty impressive list. It seems like the study is adding to the growing body of evidence suggesting that delaying reproduction could benefit both lifespan and health span.
Absolutely. To validate their findings, they analyzed data from nearly 200,000 individuals in the UK Biobank and found similar results.
Interesting.
Women who experienced menarche before age 11 or gave birth before age 21 had a significantly higher risk of developing these age-related diseases.
That's pretty compelling. It certainly adds another layer of complexity to our understanding of the aging process.
It does.
We've covered a lot of ground already, but we have One more fascinating study to discuss before we wrap up.
I'm eager to hear about it.
This last one, published in bioRxSorv on January 25th, takes us deep into the world of cellular cleanup.
Okay.
It's by Du and their team from Zhejiang University. And it focuses on how our cells deal with protein aggregates, those clumps of misfolded proteins that can cause problems.
Ah, yes. Protein aggregates, they're like gum in the machinery of our cells.
Right.
And this study sheds light on how cells clear them out during cell division, a kind of spring cleaning for the newly divided cells.
Well, it's like a fresh start for the daughter cells.
Exactly.
They inherit all the good stuff, but not the accumulated junk.
Exactly. And what's really interesting is the specific mechanism they uncovered for this cleanup.
You okay?
It involves a chaperone protein called BiP, which helps to unfold and refold those misfolded proteins, giving them a chance to become functional again.
So BiP acts like a cellular repair crew, fixing the damage.
Indeed.
That's pretty amazing.
And they found that this cleanup process is especially active during a specific phase of cell division called mitotic exit.
Okay.
It seems that cells take extra care to ensure their offspring start with a clean slate.
This has some pretty intriguing implications for aging, wouldn't you say?
Absolutely.
If this cleanup process becomes less efficient as we age, it could contribute to the buildup of protein aggregates, which we know are linked to various age-related diseases.
Absolutely. This research underscores the importance of understanding how cells maintain proteostasis, that delicate balance of protein production, folding, and degradation.
Right.
It's a key area of focus for developing interventions to combat aging.
Well, we've covered a lot of ground today, from autophagy to proteomics to the intricate workings of our cells. It's clear that longevity research is advancing at an incredible pace.
It's a truly exciting time to be in this field.
Yeah.
The diversity of approaches being taken to unravel the mysteries of aging is inspiring.
We've seen researchers using cutting-edge technologies delving into the molecular details of our cells and even taking a holistic view of the body as a network of interconnected systems.
Yeah.
It's a testament to the collaborative and interdisciplinary nature of longevity research.
For sure.
And with each new discovery, we're getting closer to understanding how to not just extend lifespan, but also enhance health span, living longer, healthier, and more fulfilling lives.
Absolutely.
As always, we encourage you to delve deeper into any of the papers that sparked your interest.
Definitely.
The links are available on longevitypapers.com.
And if this deep dive has left you pondering the future of aging research, here's a final thought. Imagine a world where aging is no longer seen as an inevitable decline, but rather as a process we can understand, modulate, and even reverse.
It's a bold vision, but with the rapid pace of scientific discovery, it may be closer than we think.
I agree.
Until next time, keep exploring the fascinating world of longevity research.